You are here because you want to bring to patients an engineered cell-based therapeutic. For most of this audience, that's going to mean engineered T cells for CAR T. That's your 800-pound gorilla in the cell therapy space. CAR T's out there, it's working. We see FDA-approved therapies, so we'll be giving that tech plenty of screen time today. In the CAR T space, what challenges are you facing? You're looking for improved persistence in the face of T cell exhaustion. You want better fitness in the context of the tumor microenvironment, thinking in particular about overcoming those immunosuppressive properties that cause immune resistance. You're thinking about safety and limiting potential side effects and toxicities like cytokine release syndrome. You're wondering how to get around antigen escape, maybe by finding new antigens to target or making better use of the targets that we already have. You're working on T cell trafficking, getting those cells to the tumor, or on tumor infiltration. And those are hard problems, to say the least, but we think that Ginkgo is well positioned to support you here because a lot of these problems come down to cell programming. You want to optimize the CAR domain itself, maybe add some new domains to the design, maybe make some additional modifications to the cell line. I am not a corporate spy. I don't know what types of clever designs your R&D team might have cooked up, but I bet you that they are likely to benefit from the kinds of large-scale, data-driven combinatorial optimization that we do well here at Ginkgo, because that's just the nature of complex biological problems. You need to screen large numbers of variants to hit on the best designs. You need big data sets to extract the best design rules. And personally, I can't imagine a cell therapy strategy where you wouldn't want that. And I think we're going to show you some data today that will convince you that the combinatorial screening strategy can be effective, at least for one particular type of CAR design. We're also going to be looking outside of the CAR strategy. What about other cell types? NK cells or macrophages? What about other disease areas like autoimmune or wound healing? We're going to try to give you the broadest possible sense for our capabilities so that you can think about how foundry scale engineering for those applications too. We can't do everything yet, but the aspiration here is to offer a set of services to reprogram any cell in any way. So if you're working on a cell therapy, it's probably in scope for us. All right. So let's get to it. We are going to start with a brief introduction to Ginkgo, who we are and what we do. Then we're going to hear from our technical team on our capabilities in cell therapy. First off, we're going to get an overview of our mammalian cell foundry. Then we're going to deep dive on CAR T. We'll show some recent data from a CAR project that we did, give you a taste for the kind of cell programming at scale that we can deliver. Third, we'll take a look outside of the classical CAR paradigm. What else is Ginkgo bringing to the table in cell therapy? We'll touch on iPSCs, gene editing, and inducible promoter tech. And then finally, we're going to end with a nice rich round of Q&A. And to that end, I would like to call your attention to the Q&A feature somewhere at the bottom of your Zoom window. That feature is live. You can click on that button at any time to ask us a question. And I am pretty excited about this Q&A session because we've got some serious talent on the line. On the business side, we've got Yech Kang. He can answer your questions about how to partner with Ginkgo. On the technology side, we've got our two featured speakers, Shadi Eshki, Taeyoon Kyung. They have been extremely close to this work over in the Mammalian Foundry. Plus, we're bringing out our VP of Mammalian, Narendra Maheshri, who I think many of you know by reputation, giant in the field, mammalian cell engineering. I am a student of the webinar format, a humble student. But one thing I've learned is that people want to hear from thought leaders. Okay, so that's who we're bringing out today. We're going to give you the best possible webinar experience because here at Ginkgo, we believe in customer service. Speaking of which, let's talk about Ginkgo. We are over 1,200 people. We're based in Boston. We have offices in California, France, Netherlands, Switzerland. If you come to visit our headquarters, you'll see a lot of robotics and automation. That's our Foundry platform. Between the technology investments and the physical scale, we've invested almost $500 million in building out this foundry. We didn't do it so that we could develop our own cell therapies. We did it for you so that we can partner with you to make those products. A good way to think about our business model is we're a CRO. You can send us your R&D projects on a contract basis, but we want to be more than a traditional CRO in the sense that you can come to us and get things that you can't get anywhere else. Part of that is all the automation infrastructure and the expertise around cell programming that we've built out. You get access to a platform. It took many years and many millions of dollars to build out. Start using it right away instead of taking the time to build it yourself. Part of that is that we have the flexible capacity to execute your project at the level that you need for the place where you are in the R&D process. So if you're doing heavy R&D work this year, you can spend more. If your projects are wrapping up next year, maybe because you're going into clinical trials, you can spend less. Okay, so that's the sales pitch. Let's get to the tech. First up, we've got Shadi Eshki. Shadi is our Senior Director of Mammalian Engineering. She has a PhD from MIT, BioE, where she worked with Linda Griffith and Harvey Lodish. She was a senior scientist at Caribou Biosciences before coming to Ginkgo, where she has been hard at work for the last 3 years, building out our mammalian cell engineering capabilities. Welcome, Shadi.
Thanks, Jake. It's great to see everyone here. I'm going to share my screen. Okay, so in our format today, we're going to start with an overview, a very high-level overview of what we do in the mammalian foundry, and then we'll go into a deep dive on the CAR-T proof of concept work the platform that we've built so far, and end with 3 sort of snippets of our emerging platforms in adjacent spaces. And I'd like to start with some really exciting work from outside Ginkgo, demonstrating how advances in synthetic biology are revolutionizing medicine. We've seen this crowd knows quite well the story of Emily Whitehead and the remarkable success that autologous CAR-T for blood cancers has experienced. But there's other areas that really are inspiring to me personally. AAV therapy, curing blindness in children, CRISPR-based therapies that are really restoring quality of life to sickle cell patients, as well as true organ replacement in type 1 diabetes with the new pancreatic cell products. And while these are all very exciting clinical developments, I actually consider them as really proof of concept studies. You know, is it even possible to deliver a therapeutic gene with a viral vector systemically and see a therapeutic impact? Is it even possible to gene edit somebody's cells and then deliver them back into the body, et cetera? And as Jake pointed out, our goal at Ginkgo is not to build more of these products and take them to the clinic, but rather to provide you and other therapeutic developers with the tools to optimize these. And so that includes increasing the efficacy. You know, as we all know, these kinds of products don't work in all situations for all types of disease. How to make these more safe, and importantly, how to increase access to what are actually very expensive advanced medicines. And our mission here at Ginkgo is to make biology easier to engineer. And we feel that the kind of systematic high throughput experimentation that we do will be able to deliver more of these products to patients. And so that's a little bit about the overall motivation of our mammalian foundry here. And in the last 4 years, we've been able to build out our high throughput cell engineering platform into 3 business verticals. These include RNA therapeutics, cell therapy, which is what we'll be talking about today, and AAV. And one way to think about the Ginkgo infrastructure is really a platform of platforms. We use automation and robotics in all of the work that we do. And as we enter a new space, we build a sort of specific version of that high throughput platform for the individual use cases. So for example, what that means for AAV might be the high throughput transfection process and titering process that we've put together, the capsid assets that we've brought in, the manufacturing work that we do. So these all leverage the same core approach, the same core instrumentation and infrastructure, but they are now highly specific to the problems at hand. And so what these areas all share in common is high throughput exploration of design space. And in the next slides, you'll hear a story of that specifically and how we're deploying this platform for CAR optimization. So I will stop here and hand it over to Yoon.
Okay, thank you, Shadi. Now we're gonna take a closer look into what Ginkgo's Foundry can deliver in practice. We're gonna deep dive into a particular case study of a car engineering project that we recently executed, and for this, we're turning to the guy who knows the tech probably better than anyone, and that's Taeyoon Kyung. Taeyoon is senior mammalian engineer. He has a PhD from KAIST, Korea Advanced Institute of Science and Technology, postdoc at MIT, worked in industry optimizing car architectures before coming to Ginkgo. Welcome, Taeyoon.
All right, thanks, Jake. Let me first share my slides. All right, thanks, everyone. Really excited to share our CAR-T story with you guys today. So first of all, let me start off by explaining the library design that we built here at Ginkgo. So as you can see here in the scheme, we have 100 different types of signaling domains that we are randomly incorporating into one of these 3 positions in our backbone of the library. And these 100 different domains are coming from items, which are core T-cell signaling domains. We have some co-stim domains, some of the inhibitory domains, and many of those domains are not just from T cells, but from many different types of immune cells, such as B cells or macrophages. So the idea here is that we wanted to approach this in an unbiased way to pull different types of signaling domains in context of T cells. So you could imagine that some of the domains from different type of immune cells could work in T cells and maybe even better than what's being used in the clinic right now. So if we incorporate into these 3 positions, that makes 100 to the 3rd, which will make about 1 million member library. If we incorporate just 2 positions, that's going to make 10,000 member library. And these 2 and 3 ICDs are grouped together just to show you that each of ICD has its own unique DNA barcode. And then, these DNA barcodes are reverse order of our ICDs, and that's just due to how we designed our library. But at the end of the assay, we can just read these barcodes to find out what kind of CAR containing T cells have survived and proliferated in our assays. All right. So yeah, what are we doing with the library? So here, the major problem in the field is that CAR T cells are not persistent enough against solid tumors. So they get exhausted, become dysfunctional rapidly in the tumor microenvironment. And the idea here is by exploring different combinations of signaling domains, we are trying to find combinations where they could maybe resist some of those exhaustion, immunosuppression, and then proliferate and even survive in those kind of environment. So here we are using a 10,000 member CAR library. We make them into lentivirus and then transduce primary T cells. And then we co-culture those CAR T cells with tumor cells. But we don't just do this once. We do this multiple times. So for example, every 3 days, we introduce fresh tumor cells into the culture over the course of 3 weeks. So here we are trying to intentionally drive these CAR T cells into exhaustion. And during that process, can we find CAR T cells that could maybe survive and proliferate in this type of environment? So at the end of the assay, we extract genome DNA and then sequence barcodes to find out what kind of CAR-containing T cells have survived and proliferated. So this is a data set from the pooled screen where you're looking at fold change of each replicate in the X and Y axis. And each dot here is representing individual CAR combination. So first of all, you can see 2 plots here where we use 2 different libraries in parallel. So same ICD library that's conjugated with high affinity binder and one with the low affinity binder. And I'll note that the difference in the affinity between these 2 binders are about 30-fold in KD. So you can see that about 38 clones in the upper right quadrant are coming out as hits with the high affinity binder library, and then 55 with the low affinity binder library. What's interesting here is that the BBZ CAR, which is widely known, used in the clinic right now, came out as hits in both cases, but not the 20Z CAR. So you can see the 20Z CAR with the high affinity binder have not enriched at all. So what this tells us is that the potent signaling domains of CD28 and CD3 when they are conjugated with the high affinity binder in the extracellular side, that just drives them into exhaustion much faster than the other clones here, that we don't see it as a hit with the high affinity binder. But with the low affinity binder, we actually do see it as one of the hits, which is interesting. Also, I'll note that there's very few shared hits among these 38 and 55 clones here. So what that tells us is that you might need specific combination of signaling domains for specific affinity of the binder. So you can just use same signaling domain combination for every binder. It doesn't seem like it doesn't seem to work that way. You need optimization for each cases. And this is another way to look at our data set. So here you're looking at the every combination of signaling domains that are in our library. So y-axis are list of ICDs in membrane proximal position, which is in ICD1 position here. And then x-axis is ICDs in membrane distal position, which is here. Oops, sorry. So in this case, you can see the upper left quadrant. You see a lot of red dots here, which means they are enriched a lot. But this is kind of expected, given the fact that these are composed of activating and co-stim domains of T cells. So you would expect them to survive and proliferate in this kind of assay. What was not expected is down here. So these are combinations of those T cell domains together with the domains that are not from T cells, but from different types of immune cells. So for one example, the one that I highlighted in the bottom here. So in this case, so by the way, this is from a B cell. This is a B cell molecule. When this is incorporated in membrane proximal position, whatever comes in the membrane distal position, there's a high chance that this CAR construct will work well in our assay. So we call this as a universal ICD. And by only using this kind of unbiased approach, we can find this kind of unexpected findings. So far you looked at full screen data sets, but the full screen is actually the beginning of our story. So as you could imagine, since cells are all pulled together, it's really hard to know single CAR design level function. Also, I'll admit that you may see some false positives just because of paracrine signaling in those pools. So this kind of array secondary screening is really valuable and important. And throughout the array screen, now we can really look at single CAR design level cytotoxicity, persistence, cytokines, different types of phenotyping, and also transcriptomics. So we have individually synthesized 100 different types of CAR designs that came out as hits in the pool screen. We ran the array screen. Here, just for the simplicity, I'm showing 10 representative CAR designs. As you can see, these are combinations of many different types of protein family members. And as you can see on the right, you're looking at the pathways that are turned on after the antigen engagement with these CARs, 10 different CARs. And you can see that they're quite drastically different, which indicates that they all have pretty much different mechanism of action when they're engaged with the antigen. And I'll also note that CAR4 design here is top of the hit in our array screen, as you'll see in the next couple of slides. What's interesting about this is that if you look at the membrane proximal position ICD, this is actually one of the ITAM mutant version of signaling domain. And the membrane distal ICD is one of the TNF receptor superfamily members. So this is actually the reverse order of what people usually use for CAR, right? BBZ, 20Z CAR, they all have CDC zeta in the distal position and then costim in the proximal position. But here we are looking at the reverse order. And that just came out from our pool screen where we approached this in an unbiased way for randomly incorporating these ICDs. And we think this is a very interesting finding. So here on the left, you're looking at the cytotoxicity of each CAR design in re-challenge. So every time you see the downward of graph, that means tumor cells are getting cleared. So fresh tumor cells are getting introduced and then getting cleared. And this went on over the course of 20 days. As I mentioned, CAR4, as you can see, they're clearing out tumor really well up to day 20 here. And then 20Z and BBZ, which are two FDA-approved designs in the field, are also performing pretty well. But for example, CAR1, you can see that in the later stage, especially, they are suffering in terms of clearing out these tumor cells. And then also if you look at the proliferation data set down here, you can see that CAR4, which is in green, are proliferating really well up to day 70 in this type of re-challenge assay. But then 20Z falls out quite early in the stage, but then BBZ in the later stage. And this is also consistent with the previous findings in the literature, where BBZ shows better persistence compared to 20Z CAR. And all these features without producing excessive cytokines. So if you look at the CAR4 here, you don't see much difference in terms of cytokine secretion compared to other CAR designs. So, for example, in terms of safety, we could say that this CAR4 outperforms other CAR designs without compromising the safety. So you're looking at the more challenging assay format here, and just comparing CAR4 and BBZ here. So against Ginkgo cell line, which is a hematological cancer, so this is a lymphoma cell line, you can see that both BBZ and CAR4 clear out tumor really well. But if you look at the A549, which is a lung cancer cell line, BBZ is starting to suffer clearing out tumor cells, as you can see here, but not the CAR4. They're controlling tumors really well. If you look at the graph here, y-axis is looking at the tumor cell counts. As I mentioned, CAR4, which is in green, is clearing out tumor much better than BBZ in these two serial re-challenge assay. But the place where we see the most advantage of CAR4 design is the proliferation capacity of these T cells. So you're looking at the self-trace dye experiment across 3 different tumor cell lines. So A549 and MDA, these are solid tumor cell lines, and Ginkgo, as I mentioned, this is a hematological cancer cell line. And since this is a self-trace dye experiment, you're looking at the dilution of the dye, which would happen every time cells divide. So more the dilution is, better the proliferation of these CAR T cells. So if you look at Ginkgo data set first, so both BBZ and CAR4 seem to proliferate really well. CAR4 a little better than BBC over the course of day 6. But again, solid tumor cells here, so breast cancer and lung cancer cell lines, you can clearly see that CAR4 is proliferating much better than the BBC cells, especially BBC is having a hard time proliferating against A549s here. And then if you look at the high tumor burden assay data sets, this is also interesting. So at E/T ratio of 1:1, so this means CAR T cell and target cell number is the same at 1:1 ratio. You don't see much difference in terms of tumor cell clearance here. So CAR4 is basically clearing out tumor as much as 20Z and BBC. But when we look at E/T ratio of 1:8, which is a much harder high tumor burden environment, you can see that CAR4 is definitely clearing out tumor much better than 20Z and BBC. And this tells us that CAR4 is probably not clearing out tumor faster. So if you look at the single cell level resolution, CAR4 is probably not clearing out tumor faster than BBC or 20Z. But they can proliferate much better, which accumulates competitive advantage over the course of days. That it does seem like they're clearing a tumor much better than the other designs. And then all these phenotypes, functional phenotypes, without compromising memory and exhaustion phenotypes, as you can see here. So first, on the left, so red bars here are central memory phenotype cells. And as you can see, with the CAR4 design, you can see larger proportion of central memory T cells compared to 20Z and BBC, but less of effector memory phenotype cells, which is in green. Also on the right, we are looking at PD1, TIM3, and LAG3 stained cells, especially if you look at the triple positive ones here. So BBC and 20Z, they are a little less of 40% that are exhausted and got stained with these 3 different exhaustion markers. But then CAR4, you can only see a few percent that are positive with all these 3 markers. So basically, CAR4 is proliferating and killing tumor cells at high tumor burden much better than the other designs without compromising the memory phenotypes and also getting less exhausted compared to the other designs. This is the last data set we have here. So here we try to make this assay even more physiologically relevant by adding combinations of TGF-beta and adenosine. So you're looking at different concentrations of TGF-beta and adenosine in this 4x4 matrix. As you can see, in all conditions, CAR4 cells are basically surviving much better than BBC cells. So this implies that CAR4 signaling domains, its CAR4 design can resist some of this immunosuppression that's coming from TGF-beta and adenosine. And lastly, just wanted to share that so we just showcased our ICD library, but that's not all we are looking at right now. We can also easily tune different types of extracellular components in the CAR. So we can basically explore different types of binders with different affinities. We can try out different hinges and transmembrane domains. Also, we can optimize bispecific CARs, for example. And then also we can optimize synergy between these CAR signaling domains and the payload we can express together with the CAR, such as cytokines, switch receptors, and also the cloaking factors, which Xiaoli will explain a little further in the later slides. So yeah, this is all we have so far for the CAR engineering field. And we are, excited to work with you with all these platforms. And back to you, Jake.
All right, thanks. Thanks, Yoon. So while I've got you here, I want to, I have a quick follow-up question. So I'm wondering, what is your sense for the diversity space for the ICD domain. So you mentioned that this was a 10,000 member library that we pulled out CAR for. Is that a good coverage of the design space? Could we do 100,000? And if we did, what would be the benefit of that? Could we go down to 1,000? And what would we lose from a smaller screen?
Yeah. So I mean, first of all, like if we go down in terms of a diversity scale of our library, we have less chance of finding good cars. Basically, that's the downside. And then if we could certainly increase the diversity of our library. So for example, you mentioned 100,000. So 100,000 car designs is still very, very much manageable. So you could imagine combining these 10,000 member library, ICD library, together with 10 different binders, maybe 10 different hinges or transmembrane domains. So in that way, you could optimize extracellular part with the intracellular part all together. Maybe you could find even better CAR design than CAR 4, for example. Yeah.
What does your gut say? Do you think that it's out? You think it's out there? You think there's still more space for the optimization? I know you don't know, but I want to make you say anyway.
Definitely. I mean, yeah. So far, like if you look at the literatures, it's always optimization of just extracellular part or just intracellular parts. There have been really no instance where people try to optimize both things all together. So yeah, in that space, like we could easily jump in and try to optimize those two parts all together to find the most optimized CAR design. Yeah. Interesting.
Very cool. All right. Thank you so much, Taeyoon. So now we are going to throw it back to Shadi. We've got a kind of a lightning talk format, spotlight talks on just a survey of some of our other capabilities in the cell therapy space. We're going to try to get through them quickly here, just to give you a sense of everything that we're capable of here. And with that, I throw it to you, Shadi.
Thanks, Jake and Taeyoon. So yeah, as Jake pointed out, we wanted to kind of expand the offerings that you walk away with for what our platform is capable of. And in particular in cell therapy, I think we're at kind of an interesting time in the evolution of this modality, this therapeutic modality, where, autologous works clearly in the indications it's been tested. But as you all know, it's far from an ideal therapeutic given the challenges with manufacturing and preconditioning the patients. And so there's a number of other ideas that are being tested, from allogeneic donor-derived cells, which is something that I worked on in my previous role. And so here there's a different set of challenges and opportunities. There's also iPSC-derived products, and again, a different set of challenges and opportunities, and again, engineering challenges that we are addressing in our work. And then, interestingly, there's sort of this rise of the in vivo programmed cell therapy, and how maybe you can just deliver the CAR and the other reagents directly in the body. And so here at Ginkgo, we are excited about all of these therapeutic modalities, and importantly, we're agnostic to which one of these will be the winner, right? And we're eager to work on all of them with our partners who come from across these spaces and these backgrounds. And we have built a generalizable set of services to address all of these challenges and opportunities. And some of them are shared. For example, if you need to deliver a CAR to a solid tumor, you're going to need a new binder. And maybe that's something that's common across these areas, or maybe you need to optimize the ICD. But for example, for an allogenic cell therapy today, that really makes gene editing obligatory. And so how can we optimize the guide RNAs and the off targets and even the development of novel gene editing tools? These are all things that we're actively working on here. And in the next couple of slides, I want to spotlight a couple of those areas specifically. So for iPSCs, this is something that is close to my heart, having done my academic work in this field, and something to me that appears as a real fundamental challenge to increasing adoption of iPSC-based therapies is the need for immune cloaking. If you're going to make an organ that's going to be implanted into a patient's body for hopefully decades, it needs to coexist with the host immune system. And similar to the kind of success stories about AAV therapy and CAR-T, I think that there are some promising proof of concept strategies in this space, where individual pathways of the immune system have been modulated through knockouts and knock-ins to show pretty good protection in NHP models. But again, we really consider this as a first generation type product. And here at Ginkgo, we really have the opportunity to learn from the many biological systems that currently have very good ability to hide from the human immune system. So this includes bacteria, viruses, cancer, the maternal-fetal interface. All of these systems are rich with proteins that confer an ability to hide. And here at Ginkgo, we have a lot of strengths in metagenomic sourcing, protein engineering, and high throughput immune cell assays that we feel very confident that we can discover some new approaches to the immune evasion. And so this really requires these core competencies in all of these areas. And what it would enable on the other side is sort of a comprehensive, sort of broad spectrum approach to immune evasion, and not just maybe blocking an individual pathway, but coming up with a strategy to shut all of them down. And that might very well be tissue specific. So what's needed for a beta cell replacement therapy might be very different from what's needed for a neuronal replacement, right? So again, this is an emerging area for us, and we really feel like we have a lot of the unit operations in place through the library design and the screening to really be impactful here. And so definitely, this is something that you're working on for your products. We're certainly very interested to connect. Okay, another quick vignette for an iPSC relevant topic is around knocking in these therapeutic cassettes in a stem cell, and then the problem of differentiating them to the cell type of interest. And often what is seen is silencing of that therapeutic transgene. And again, you know, to have that therapeutic impact, you really need the payload to be present. And so we see this as there's really two major parts to this problem. And one is the genomic location of the knock-in and the context, the sequence context that can affect the expression strength and stability. And one of the other major players there is, of course, the promoter, and how the two of those choices can impact the outcome. And so here we're undertaking again, a large screening campaign where we're knocking in different combinations of promoters and cassettes all across the genome, and then selecting cells based on expression strength at different differentiation checkpoints, and then recovering the insertion sites and using that, recovering not just the insertion sites, but also the promoter sequence, and using that as a framework for sort of more rational and site-specific engineering strategies. So that's, again, another tool in the iPSC toolbox. And then finally, really getting at one of the other fundamental problems for iPSC-based therapeutics, which is the very lengthy and incomplete differentiation processes that are required to go from a pluripotent cell to a differentiated effector cell type. And here we have a number of offerings. These include automated DOE approaches to identifying the best combinations of exogenous factors to optimize an existing differentiation process, as well as screening libraries of transcription factors to discover a new differentiation process. And in particular, for that latter point, we do believe that in the long term, moving away from a biochemical process and all of the variability and the cost of goods inherent in that, as is currently done with exogenous factors in iPSC-directed differentiation, moving away from that to a genetically controlled process will allow shorter timeframes and more robust cells out at the other end. Okay, and then moving along, wanted to also spotlight our work in gene editing. This is, again, another area that I'm really excited about, having worked at sort of a CRISPR 1.0 company in the past, and really just excited by all of the capabilities that we have under one roof here at Ginkgo to address these challenges. And the idea here is that Ginkgo has a very large proprietary collection of metagenomic sequences. And while we're mining these already now for a variety of natural products and other gene clusters there's also the opportunity to look for gene editing enzymes. And I think the common theme that hopefully you'll take away with you today is that our ability to not just do the bioinformatics, but also to screen these putative gene editors in very high throughput is something that we're good at and we're proud of. And so in our mammalian cell engineering team, in the last few years, we've been able to onboard over 60 different cell types, including primary cells and iPSCs, as I just mentioned. And so our ability to test these gene editors in your therapeutic cell of interest is unparalleled. And as a case study for something that occurred a long time ago here at Ginkgo was that we actually undertook a new DNA binding enzyme engineering campaign to improve our in-house DNA synthesis platform. And in that work, we were able to synthesize and screen 30,000 different DNA error correcting enzymes, and we're able to select variants that had improved performance, and these are in use in the internal process today. And so now building on that work, we're really focused on large serine recombinases as useful genome engineering tools. And there's been a lot of interest in these, both in combination with DCAS9-based prime editing strategies, as shown on the left, on the right, sorry, there, as well as potentially on their own for integration into the human genome. And of course, the perhaps most well-known application in sort of the landing pad engineering type scenario. And so as part of our acquisition of Zymergen last summer, we also got access to their metagenomic database. And an initial bioinformatics search of the databases revealed that we have over 80,000 putative serine recombinases in our collection. And just compared to what's publicly available in GenBank, it's over tenfold more. And importantly, that GenBank collection was the source for a recent publication that demonstrated the discovery of a number of very promising recombinases that have specificity for endogenous human sequences. And so this is something that we're really excited to initiate right now. And so that what that entails is really going, actually synthesizing these sequences and then screening them through a variety of biochemical and mammalian cell assays. So again, if there's a particular class of gene editing enzyme that you feel is necessary for the kind of engineering work that you're doing, or maybe it's a simple FTO kind of problem, which I know in this field is quite common. We are very excited to do this kind of work in a partnered way. So I'll skip over this one that demonstrates the nature of the recombinase sequences. Again, very happy to follow up on that. And then finally,
Shadi, can I jump in here?
Please, yeah.
Sorry, I just in the interest of time, I want to make sure we have some time for Q&A.
Great.
So maybe we'll skip over that last section, because I want to make sure we have time to open up the Q&A to the audience here. And for this section, I'm going to call your attention to the Q&A feature at the bottom of your screens. You can type your questions in there. We'll be doing our best to get you an answer. For this, I'm bringing out the full team here. So on the business side, we've got Yech Kang. He is focused on our therapeutics partnerships. He can answer all your questions on the business side. On the tech side, we're bringing out Narendra Maheshri, VP, Head of Mammalian Engineering. And I want to open with a question for Narendra. So we've heard about a lot of different capabilities today, but we know that we don't want to just offer our customers like a pile of different services. We want to do real R&D, and we know that R&D needs to be integrated. So how does Ginkgo's Foundry integrate these capabilities to deliver a finished R&D project?
Thanks, Jake. So I think you can view Ginkgo as really a research collaboration partner. So we like to discuss with a potential partner what is the problem or challenge they're really trying to tackle, and there are probably a number of pieces to tackle that problem that we already have. Maybe a part of it is CAR. There's a question about CAR NK as an example, right? We have a CAR library. We have folks who've worked a lot in NK cells. We haven't done a screen in CAR NK. We'd love to work with a partner to do that, and the sort of assays, et cetera. Those are things we are really able to rapidly develop, even, for example, in a cell type where we haven't perhaps done it before. So you should think of us as really good integrators of multiple unit operations, as Shadi was mentioning, to deliver something for your specific problem. And we're always open to building something or investing further in something new, if that makes sense for our partners, whether that's equipment or capabilities or something else.
Mm-hmm.
Don't know if I answered whoever's question that was. Happy to elaborate.
Yeah. I think so. I got another question on the business side. Maybe this one is for you, Yech. So what is your business model? What kind of licensing structure can I expect from Ginkgo?
Yeah, that's a really good question. So we have a pretty flexible model, and we do try to make an effort to tailor to each of our partners' needs. With that said, you can really think about like two key buckets of partnership structures. The first is, as Narendra alluded to, it's our typical research collaboration and licensing model. So these are highly individualized projects, will look very similar to the case study that Yoon ran through today in his presentation, where we'll have multiple discussions to align on the scope of work, determine what your target specifications are, as well as the final deliverables. Typically, the end deliverable is a final design. So an example can be an optimized car ICD domain, where you'll have the opportunity to license that, and it'll be fully optimized for your needs. The second bucket is there's sort of a group of partners that we work with that want to basically test something in their hands today, something that works. And so as you've seen in our presentation, there's a number of things that Ginkgo has validated internally, whether it's car ICD domains, promoters, regulatory elements. So what we can actually do is we can allow you to test a small subset of these in your hand. We call this an evaluation and licensing model, where at the end of a pre-specified evaluation period, you'll have the option to either one, license it because you like it, or two, we're always happy to engage in a discussion where we can talk about a broader partnership, right? This is a very high level description. There will certainly be nuances. And if you want to discuss specifics on IP, downstream economics, et cetera, all of which will be affected by the scope of the project, we're always happy to connect in a follow-up conversation after this.
Jake, you're muted.
Oh my God. Oh my God. Lightning round questions. For Narendra, have you validated your car constructs in vivo?
We're currently validating some select hits in mouse models, and we're also gearing up with our collaboration with Wisconsin. So this is with Chris Saha and Christian Caputini there to do a pooled screen in mice with a anti-GD2 CAR. So yeah, stay tuned.
Very cool. Quick one for Yoon. Can you work on binders and other CAR domains?
Yeah, I mean, I just showcased our ICD library with 2 different binders with different affinities. But in theory, maybe we could try this out with 10 different binders or even 20. The idea there is, so there are 2 approaches, right? One where you could pull everything together into one bucket and try to find the most optimized version of car design. In the other scenario, which is what we did here, you could have same ICD library with multiple different binders in the arrayed format for the screening. So this is both pooled and arrayed format screening at the same time. But the idea here is trying to find ICD hits from each affinity binder first to see if there's any shared hit. If not, it's a very biological question, right? Like what kind of a signaling domain combination could benefit from each bin of affinity coming from binder? So this question for the first answer for the first one, but then for the second question, we could certainly increase our ICD pool. So we just use 100, but it could go up to 150, 200, depends on our partner's need. But also, like I mentioned, we could vary the hinge domains or transmembrane domains, some other components of the car, all at the same time.
All right. Thank you so much, Yoon. And with that, the time has come to wrap up this virtual event. Thank you so much for joining us. Thanks to our team who came out today, Shadi, Yech, Yoon, Narendra. After this call ends, you're going to get automatically sent over to a survey. This is the easiest way to know how to follow up with you. Let us know if you're ready to start the conversation, and somebody from Ginkgo will reach out. Also, let us know if you've enjoyed this event and found it useful, because I love doing these and I'm excited to make them the best that they can be. One last thing, if you joined the call a bit late, maybe you missed it, but we opened a little poll on the very urgent question of what is the correct emoji to represent a mammalian cell? And now the results. The answer is egg. The answer is egg. So that was surprising to me, but far be it from me to disagree with the consensus of this community. It's clear consensus. The right emoji is egg. And with that, I will share our call to action. The call to action today is cell therapy at ginkgobioworks.com. This is one of many ways to reach out to us. If you want to get started, put the power of the foundry behind your project. And until next time, as we like to say around here at Ginkgo, let's grow.